Good day, everyone. It's funny, I think I'm wearing the same shirt that's in this picture, even though this picture was taken a year ago. Um, so I'm going to be talking about silicon as a fertilizer and our work with enhancing stress tolerance of bedding plants and potted plants. So begin with what is silicon. So silicon is an element. It's actually the 14th element on the periodic table. And if you remember your chemistry, it's right below carbon on the periodic table. So it's a very flexible element in that it wants to form four bonds with other elements. Silicon is the second largest constituent of soils. So a lot of our soil structure particles are aluminosilicate materials. So soil by weight is about 47% oxygen, 27% silicon, and 8% aluminum. So plants are growing in an environment where there is a lot of silicon around. And I want to point out that for the purposes of this talk, we're talking about silicon, not silicone. Silicone is a polymer of silicon, so carbon, hydrogen, and oxygen connected to many different silicon atoms. It has a rubber-like consistency, and it's commonly used in cookware, lubricants, adhesives, and so on. But we're going to talk about silicon, so the element silicon, and then the form that plants take up is orthosilicic acid. So that's silicon connected to 4 oxygens connected to 4 hydrogens. Once plants take up silicon, there is deposition in their shoot tissues, and it's typically deposited as silica. So that is silicon connected to 2 oxygens, and then that is connected in a 2D or 3D type chain. It would be similar to quartz or amorphous glass. So there's been a lot of research in the agriculture world relating that plant-absorbed silicon, the orthosilicic acid, to benefits, especially for rice. Rice is about 5% silicon by dry weight, and scientists have found the actual genetics related to silicon uptake in the roots. So rice roots are actively selecting for and absorbing silicon and transporting it to the shoots. So about 5 years ago, we had the question of would floriculture species in a soilless potting mix, a peat perlite potting mix, take up silicon? And so to answer that question, we looked at 21 different floriculture species. I'll show you them in a minute. They were growing in a peat perlite potting mix, which had about 2 parts per million of that orthosilicic acid. Our tap water at Cornell also has a little bit of silicon as well, 0.8 parts per million, but we were giving the plants weekly drenches with 112 parts per million potassium silicate. We did that for 10 weeks, and then this chart shows us leaf silicon content. It would be in parts per million. So we have our 21 different species on the bottom, and then the shaded bars are the control plants that did not get supplemental silicon. So on the lower left side, we have this Petunia Cascadia cherry spark. It accumulated about 200 parts per million silicon just from what was present in the potting mix and from the tap water source. When we gave it the once weekly drenches of potassium silicate, it had significantly higher silicon in its leaves at about 500 parts per million. So an interesting thing is that our bedding plants really varied from Petunia on that low end all the way up to our Geranthemum and Terrenia on the high end. And of the species that we looked at, so this is percent increase in leaf silicon content comparing the silicon-treated plants to the control plants. And so we found that for 11 of the cultivars that we examined, they accumulated significantly higher amounts of silicon when we gave them that. So for these plants, it seems like they have that kind of underlying biological machinery to take excess silicon and to utilize it within the plant. When we measured several different plant traits at commercial maturity for these plants, we measured plant height, plant width, stem diameter, plant fresh weight, plant dry weight, water content, which you calculate from plant fresh weight and dry weight, flower diameter, and leaf width. And when we did that, so these positives showed us cultivar that responded positively to silicon, in a statistically significant way. And then the negatives represent plants that responded in a negative way. And then where there's no positive or minus, it means there was no difference. So what we see overwhelmingly is that there was very little, few species had significant horticultural trait responses to silicon. There are some examples here, like this lobelia, where several traits were affected. So we had slightly smaller flowers, slightly less wide leaves, but we had higher fresh weight, higher dry weight, higher water content, and a taller plant. So it seems like there was some benefit there. But the take-home message from our initial survey was that while many different plants accumulated silicon, there were not huge differences in what the plants looked like visually and how we measured these horticultural traits. One of the things to note, so these cultivars that are bolded, those were ones that accumulated higher amounts of silicon when we gave the drenches. And then the cultivars in gray were the ones that did not accumulate higher amounts of silicon. The interesting thing is we did see some significant differences in horticultural traits even for plants that did not accumulate excess silicon. So it seemed like in that case there could have been some benefit or differential growth even though the plant didn't accumulate a lot more silicon. So our conclusions from that initial screening were that weekly silicon supplementation did increase leaf silicon for half of the species we studied. There were some certainly variable silicon accumulating ability. Some plants accumulated more than others, and it also seemed like that differential silicon accumulation was not required to see a plant growth effect. So one hypothesis is that just a continual supply of a small amount of silicon may be important to elicit that response. And about the time that we were doing this experiment, we started reading more of the literature of silicon and found that silicon benefits especially seemed to appear when the plant was stressed, in some cases insect and disease stress. And then we're not plant pathologists or entomologists, so we wanted to look at plant abiotic stresses, so stresses imposed by, say, drought or salt or extreme temperatures. So in subsequent work, we decided to grow plants with and without silicon and then impose stresses and see if there were benefits on the plant. So our first experiment with abiotic stresses in silicon was a salt stress experiment. So we had 28 different kinds of bedding plants in a peat perlite potting mix. They were given a constant silicon supply in this case, so 56 parts per million silicon as part of their fertilizer program. And then for the salt stress plants, they received about 900 parts per million sodium and about 1,400 parts per million chloride, so high sodium chloride stress. We calculated that the cost of silicon throughout that 5-week period came out to be about 2 cents per pot using the Dynagro Protect potassium silicate. So we found for 8 of the species we looked at, the salt-stressed plants that received silicon were significantly larger than control plants that had not received silicon. And these are the plants listed on the right side: Bacopa, Begonia, Calibrachoa, down to Snapdragon. For those plants, the salt-stressed plants were 47% to 152% larger. And one example of that is impatiens. So this is garden impatiens and Impatiens wallerana. So we see shoot dry weight, our measure of plant size, and our plants that did not have salt stress, these minus sodium chloride plants, there was no difference in size between them. But sodium chloride had a huge effect on impatiens plant size. So our sodium chloride treated plants that did not have silicon were the smallest ones. Those are the ones with the letter C here. And then silicon addition definitely improved the growth of those plants. However, they were not as big as our plants that received no salt stress. But it did help improve salt stress tolerance to some degree. We did also find that adding silicon did not reduce the growth measured in dry weight by any of the unstressed plants. And there were 4 additional species that showed some benefit of silicon, but but the plus salt plus silicon treatment was not significantly different from the plus salt and minus silicon treatment. Actually, one of those that was very promising, but plants were essentially dead by the end of the experiment, was New Guinean patients. So it's interesting that impatiens showed us a nice response as well as New Guinean patients. So we have a control plant on the left that did not get salt, it did not get silicon. In the middle is the plant that had this extreme salt stress and got silicon, and it was still alive for 3 or 4 weeks into the experiment. And then we have our salt stress treated plants without silicon, and they were already dead at that point. So there are some benefits, it appears, for salt stress tolerance in bedding plants, but it does seem to be on a kind of species by species basis. Another experiment that we did, this was done by a former postdoc in my lab, Roland Leatherwood. So we grew poinsettias in a greenhouse with silicon. This was another case where we gave silicon at about 50 parts per million, just as part of the standard fertilizer regime. And then once the poinsettias reached commercial marketability, we took the plants out of the greenhouse and we brought them to a room at room temperature with fluorescent lights for 12 hours a day to kind of emulate keeping the plant in an indoor environment. And then the plants were well watered before we brought them to that room, and then we stopped watering and we came back to them and looked at how many days it took them to wilt. And our silicon treated plants lasted about 3 or 4 more days before they wilted. So here's an example of Peter Star Red that was 12 days post-harvest, and you see our plant on the right that did not have silicon is already quite wilted versus our silicon-treated plant is still turgid and looking marketable. So this could be used both to improve shelf life of poinsettias if they're not going to be watered in a retail setting, such as might happen at big box stores. It could also be used to improve kind of homeowner plant conditions in the homeowner environment as well. And then we waited till the plants were wilted. Here's an example from Peter Star, and we re-watered them, and our silicon-treated plants showed better recovery from wilt as well. And the last silicon experiment I want to show you is with heat stress. So these are petunias that were growing in a greenhouse for several weeks. And during the greenhouse stage, they again either had silicon. In this case, we gave it as once a week drench at 100 parts per million silicon, or did not receive silicon, so those were the control plants. And then we put them in a growth chamber where we ratcheted up the temperatures to either 95 degrees Fahrenheit or 102 degrees Fahrenheit. So pretty severe heat stress, and we kept that going for 24 hours. And you'll see our silicon-treated plants. So if we look at the 102 degrees treatment, all the plants are looking pretty bad, but our silicon-treated plants are more upright and showing less of that wilting. And then that's even more obvious at the 95 degree temperature, where the plants are somewhat less stressed. We definitely see less wilting and more upright plants from the silicon-treated group. And here's an example. So by 3 days after that 102 degrees Fahrenheit treatment, the plants were actually starting to acclimate to that heat stress. So they were starting to get used to it and actually didn't look as bad as they did after just 1 day of the stress response. But again, you'll notice those silicon-treated plants look much more visually appealing than those plants that did not have silicon. And an interesting phenomenon, so we looked at leaf tissue concentrations for silicon either before heat stress or after heat stress, and we found a phenomenon that was also found by researchers at Toledo, Ohio at the USDA ARS and at the University of Toledo. They found it with other stresses. We found it with heat stress, where when you impose that stress on the plant, it makes the plant take up extra amounts of silicon. And so for these petunias, whether they received, in this case, 0, 50, or 100 parts per million silicon, once we had that heat stress, they really turned on their genetic machinery to take up additional silicon. So as a scientist, this is really interesting to me. It tells me that petunias seem to be responding to heat, and one of their responses to that is to tell the roots to take up additional silicon. A little bit about the practicality of using silicon. The form that we've used is potassium silicate, which you can get as a liquid. The product that we've used is Dynagrow Protect. There's another company that makes several different silicon products that is PQ Corp, and they have a product called EggSil, which is a powdered form of potassium silicate that's water. Castle, which is kind of the gel form that you can solubilize in water. For the potassium silicates, they are not compatible with our standard fertilizers. So if we use 20-10-20 or our commercial 25-pound bags of fertilizer, the potassium silicate will cause a precipitation reaction, and that's what's being demonstrated at the bottom here. So so in a stock tank, they would not be compatible. So you either have to use a 2-injector, 2-stock tank type regime, so with that second injector in the series, or you have to do a once-a-week drench when you're not fertilizing with your standard fertilizer. So for growers that have only 1 injector in their greenhouse, that would be kind of the way to apply. And so that's why for some of my experiments, I've told you we've done weekly drenches. That was for that exact same reason. If you do have the 2 injectors, probably the best thing to do would be to supply continual silicon. And the suggested trial rate, so we've used 50 parts per million silicon if you're applying that in that constant liquid feed situation, and 100 parts per million silicon if it is that weekly drench. It is a basic material, so it does raise the pH slightly. So for example, in our experiment with bedding plants, given weekly drenches for 10 weeks, we found that pH of the silicon-treated plants was about 0.3 pH units higher than our plants that did not receive silicon. And so we certainly suggest that you conduct your own trials at a small subset of your crop at first before trialing silicon on a larger basis. There are also now, SunGrow makes a substrate line that also has silicon in it. We have not done research with that particular product, but it'd be interesting to see if there's also benefits similar to those that we found with potassium silicate. So to conclude my talk, my thoughts on silicon are that adding silicon to the greenhouse for plants growing in soilless substrates, it appears to be beneficial for certain plants under certain conditions. So it doesn't seem to be a miracle cure that improves growth and does all of these marvelous things for all plants. But through our experiments, at least, it seems to be useful for some plants when a stress is present, such as heat or salt stress. If the tap water, the fertilizer, or the potting mix doesn't already contain a continual supply of silicon, so I've seen water reports, and some waters contain a lot more silicon than others. So it could be, in some cases, that your tap water source already contains enough silicon. And then just note that caveat, again, that it's not a miracle cure for every stress and for every plant. So this concludes my presentation on silicon. I would like to thank the American Floral Endowment, the Gleckner Endowment, as well as the New York Farm Viability Institute for funding our research with silicon.